This is about experiment done to prove DNA replication is semi-conservative. DNA replication is the process of copying DNA inside a cell. Now, what does the word semi-conservative means? In daughter DNA or newly synthesized DNA, one strand is from parent and another is new in each double-stranded DNA. I repeat, in case of newly synthesized DNA, double helix, one strand comes from the parent and another is the newly synthesized strand. This is called semi-conservative, as the information in the DNA is conserved half. Okay, now coming to the who did this experiment to prove DNA replication is semi-conservative and when. As you can see, Matthew Meselson and Franklin Stahl done this experiment in 1958 to prove DNA replication is semi-conservative. Now coming to the steps of the experiment done by Meselson and Stahl. Firstly, E. coli cells. First, this experiment is done with E. coli cells, and so DNA replication is semi-conservative is first proven in case of E. coli, later in higher organisms like plants and animals. So what Meselson and Stahl did, they had grown E. coli cells in presence of 15 NH4Cl, it's written like this. Just a moment. (15) is one of the isotopes, heavy isotopes of nitrogen. We know NH4Cl is ammonium chloride. So as I said, (15) N is a heavy isotope of nitrogen. Now, why nitrogen is used in this experiment? We know DNA is made up of three basic components: nitrogenous bases, sugar, pentose sugar, and phosphorus. So nitrogenous bases, all the bases, adenine, guanine, cytosine and thymine, contains nitrogen. So once the E. coli cells are grown in presence of heavy isotope of nitrogen, that is ammonium chloride containing N14, for a long period of time, what happened that this isotope got inserted in DNA in form of bases. As a result, when we use density gradient centrifugation, that is cesium chloride density gradient centrifugation, we can differentiate these DNA from the normal type. As a result, these can be differentiated from normal DNA containing 14 N using cesium chloride that is CeCl, cesium chloride density gradient centrifugation, heavy isotope containing DNA can be easily separated from normal DNA. Now coming to the next step. What can be found as you see in this image? So first, cells will grow in presence of N15 DNA. I repeat, N15. 15 N, that is heavy isotope, shown in red color. Then after 20 minutes, that is generation 1, so half of the DNA newly synthesized, one strand will contain newly synthesized DNA or normal, and one strand will contain heavy isotope of it. Okay, now coming to the next step, how this can be obtained. Later, after few generations, later means after few generations, E. coli cultures are shifted to cultures containing normal nitrogen, that is 14 N, and they are allowed to grow. Now what is the result? We can see that DNA extracted from the first culture, as I told in this, contains half amount of heavy isotope containing DNA N15 DNA and half amount of N14 DNA. This, as you can see, it's written after 20 minutes. Why? Because the division time of E. coli is 20 minutes. Okay. So DNA extracted from the generation 1 culture contains hybrid or intermediate density. Then here middle density is the middle. In case of this one, it's heavy isotope. First step, as I told, it's cesium chloride density gradient centrifugation in the first step, As I told, it's grown in presence of only nitrogen, heavy isotope of nitrogen. So it will accumulate at the bottom, after extraction. Then after first generation, at the middle. After 40 minutes, that is after the second generation, what we can find that half amount of light, that is normal type, and half amount of hybrid. See, these two are light, these two are hybrid. So that's after 40 minutes. As you can see, after another generation, that is added 20 minutes when you add. So what happens? Compose equal amount of hybrid DNA and light DNA. Now coming to the next topic, that is difference between DNA and RNA. Difference between DNA and RNA in composition. What we know? First difference is, number 1, DNA contains 4 bases, that is adenine, guanine, cytosine and thymine. Instead of thymine, RNA contains uracil. So instead of thymine, all 3 bases are same, but instead of thymine, it contains uracil, U. And the full form of DNA is deoxyribonucleic acid. RNA is ribonucleic acid. So we can say one more. So one more, ribonucleic acid is present. As you know that a 2' OH group is present at every nucleotide in RNA. That is why it is ribonucleic acid. So 2' OH group is present. In case of DNA, it's 2' H group. So 2' OH, okay, no time. Let me finish. And DNA acts as genetic material in most of the cells, most of the living beings, and RNA is the genetic material. RNA is genetic material in only few viruses. Except then that, all other living beings have DNA as genetic material. So I repeat 3 points. Instead of thymine, the RNA contains uracil. In case of RNA, 2' OH group is present, and in case of DNA, it's 2' H. The O is not there. That's why it's called deoxy in case of DNA. And mostly DNA are double-stranded, RNA are single-stranded. DNA mostly double-stranded, means present in double-stranded form in cells, whereas RNA are mostly single-stranded in case of in the cells, present in the cells.